All right, guys. So let's go ahead and review transcription. Okay. So hopefully by now, you guys understand a couple things. All right. Okay, Micah, what's this? What's this, Micah, right here?
DNA.
Good. And where is it located?
In the nucleus.
Very good. In the nucleus. Good. Now, the whole point of also cells, they need to stay alive, and so they need these guys. Okay, they need proteins. All right. So this cell, this is a cell, that's a cell. Okay, cells need proteins to stay alive. All right. So there's something inside the cell, all right, that the cells that make these proteins. So Evelyn, okay, repeat the question for me. See, that's why it's good. Make sure to get off your phone. Okay, so here we go. Focus. Okay, so focus. All right, so cells. Remember this. Write this down in your notes right now. Write this down in your notes. Cells need proteins to stay alive. Okay, so there's something inside the cell. So I'm going to use a different color. Okay, let's go ahead and use like red, right? And there's something inside the cell. All right, and so I'm putting all these question marks. Okay, there's something inside the cells, all right, that make the proteins. So this something makes the proteins. What is that something?
Ribosomes.
Very good, excellent. Okay, so remember that for the test tomorrow. All right, the ribosomes are these things inside the cells, and you always draw them like snowmen because like they really have two parts. Okay, and so these snowmen, okay, literally, okay, are the ones that build these proteins, that make these proteins. But remember the problem. The problem, just like Micah said, is that the DNA is inside the nucleus. Okay, and so basically all the instructions are trapped in the nucleus. Okay, they're literally trapped in there. So what we have to do is we have to get those instructions out to these guys, all right, okay, so that they can make the proteins. That's the problem. So what ended up happening over time, right, was this idea, like, and so here's where in the video where it's kind of cool that I showed it to you, right? So in the video, there's this portion here, okay, where DNA, and then I think I wrote it, let me see, how did I write it? Okay, so I wrote it in here, nope, not here. Okay, so here's what I did, right? So in the video, hopefully you guys understood, okay, that DNA, and hopefully by now you guys understand, okay, and I'm getting confused because this was in the video. Okay, so hopefully in the video you guys understood this, right? Okay, Micah, how many sides does DNA have? Somebody help out Micah. How many sides does DNA have?
3.
No, not 3.
2.
2 sides.
2. Very good. It has 2 sides, right? So this right here, imagine that's the DNA that we're working with, okay? So this piece of DNA, okay, this piece of DNA, okay, really is in the nucleus. And so what we have to do is this piece of DNA, right? Piece of DNA, okay, has instructions. Okay, so this piece are basically instructions on how to make the protein. Okay, so that this, so basically this ribosome over here, right? He basically he doesn't know what to construct unless he gets this information. Okay, and then once he gets this information, then he can make the protein. And so that's the problem. So how do we get this information to the ribosome? How do we get this information out to the ribosome? Is anybody a contractor? Does anybody have friends that are like a family member is a contractor? Anybody?
Yeah, my dad.
Okay, so Anthony's dad's a contractor, right? So here's the awesome part, right? So if Anthony's dad had to build a house, so let's just use proteins and let's just say house, right? Okay, so this is the ribosome is basically your dad, Anthony. Okay, got it? Anthony's dad. So your dad is the ribosome. He's the construction. He's the contractor. So somebody, okay, somewhere made a blueprint. Okay, basically the instructions on how to build the house, right? The blueprint is so important that we got to keep it locked up, right? Because we don't want to make, it takes a lot of work to make this copy. So what do you think, Anthony, your dad's going to get to build the house? Is he going to get the original blueprint? The original instructions?
Yeah, he got to.
Yeah, well, exactly. But is he going to get the original document though?
Oh no.
No, he's going to get he's actually going to get the blueprint, which is a copy of the original plans. Yeah. Does that make sense? So you got the original plans here to build the house, right? And so what ends up happening is the contractor, he's going to get a copy, which is called the blueprint. Okay, and then that way he can read the blueprint and build the house. And so that's how your cells work. The cells work just like a construction site does. Okay, there's this architect that put together this plan, right, on how to build something, and the contractors need this plan so that they can build the house, right? In our analogy, the house is the protein. This blueprint, what is this blueprint called in our analogy? The copy. Remember, this is a copy of the instructions.
Transcription?
No, transcription is the process of making the copy. So what is this copy called? Okay, so let me show you guys, right? So here's our DNA, right? And so what we have to do, okay, what we have to do is, here we go, and I think it was here at the end, okay? So what we have to do is that DNA, okay, remember it has two sides, 1, 2. What has to happen is that DNA has to get opened up. So we have to open it. Okay, so everybody go to your virtual lab right now. Okay, so follow me along, follow along. Okay, so follow along. So in your agendas, okay, there's a virtual lab link. All right, so go to your virtual lab. So everybody go ahead and do that now. Go to your virtual lab. Okay, remember to log in so you have this. So go ahead, I'll give you guys 1 minute to log into your virtual lab. Recording? Okay, so now that you're in your virtual lab, this is the part that you guys have to understand, okay? This is the part that's tricky. So this right here, what you're looking at is basically you have to imagine, okay, that this right here, okay, is happening in here. So this is the nucleus right there. So what we're about to do, okay, is happening inside the nucleus. So this thing right here that I have in the video, it's like, imagine it's this, okay? So now the first thing that has to happen, now that we're in the nucleus, right? The first thing we got to do is we have to open up the DNA. So go ahead and open up your DNA. Open up your DNA. Okay, so essentially this is what we did. Okay, now that our DNA is open, all right, now that our DNA is open, so, and remember, we're in our nucleus, right? Okay, so here we go. Let's get that. So now that our DNA is open, remember, we're still inside the nucleus. Now what we can do, okay, with our DNA here, Okay, now we can copy this DNA. Okay, now we're going to copy this DNA. And tell me why we have to copy it again, Aaron? Is Aaron here? Okay, why do we have to copy it, people?
The ribosomes can't go inside the nucleus.
Yes, because the ribosomes cannot go inside the nucleus. Very good. So now let's go ahead and copy it. So in your lab now, okay, what you're going to use one side to make a copy of the other side. Okay, so in other words, okay, we're going to use this strand right here as the template. Okay, and this by using our base pair rules, we're going to be able to make a copy. Okay, got it? Okay, of this one. So essentially, what we're going to do is we're going to make a copy of this one, and by using this as our template, as like our thing that we're copying. So check it out. So now, if normally when you see a nucleotide with a thymine, what goes on the other side?
Adenine.
What is it?
Adenine.
Adenine. Very good. So we're going to go ahead and drag an adenine there. And so what we did is, remember this thing that looks like a little cloud right here? All right. This thing that looks like a little cloud, that's an RNA polymerase. Okay, now what the heck is that, Rodriguez? That's a protein that makes RNA. Remember what I said, that cells need proteins to stay alive? This is one of those proteins, okay? There's millions of them. So what this guy's gonna help us do is it's gonna help us make an RNA molecule, all right? So let's continue. Now, normally, if there's an adenine nucleotide, what goes on the other side normally?
Thymine.
Thymine. But remember, we're not making DNA. We're making the goal here, okay? The goal, and right now what we're doing right now is transcription. So this process that we're doing right now is called transcription. And basically what we're doing is we're taking our DNA and we're copying it and making RNA, okay? That's what we're doing right now. So because we're making RNA, we're not gonna put a thymine nucleotide, we're actually gonna put a uracil nucleotide. So go ahead and do that with me, all right? Now, the next nucleotide, okay, there's a cytosine nucleotide, so the protein, the enzyme, is now going to add what nucleotide?
Guanine.
Guanine, very good. So now go ahead and add your guanine. Okay, now if you take a look, all right, if you compare these first three nucleotides with these first three nucleotides, what do you notice? They're almost what?
Almost the same.
Yeah, they're almost the same, except for in this one you have thymine, and in this one you have the uracil, right? And so that's because, remember, we're making the RNA so we can send it out of the nucleus, right? So that we can give it to our construction workers, the ribosome. So that's why we're using these nucleotides. RNA is kind of like our copy material, right? So now let's continue through this process, right? So there's a guanine here, so you would put a cytosine here. There's an adenine here, so we're going to put not a thymine, a uracil, because we're making RNA. And so go through this whole process right now, okay, and build your your RNA. So go ahead. I'll give you guys 30 seconds. So now, hopefully you guys got a little bit of time to do it. So Annalise, walk me through it. So what am I going to add here?
A guanine.
A guanine nucleotide, very good. So let me go ahead and add my guanine nucleotide, good. And then now?
Adenine.
Good, an adenine nucleotide, then?
A cytosine.
Very good, and then?
Another cytosine.
Another cytosine, very good, okay, good, all right. And then?
And a uracil.
A uracil, exactly, that's right. Because remember, we're making RNA. Okay, and now?
An adenine.
Adenine nucleotide, very good. And then?
Guanine.
A guanine, very good. Now, if you guys take a look, okay, and compare the two, all right, this and this are basically almost basically identical, except that in here we have thymine and here we have uracil. So now what we basically have done is this guy right here that we made, this guy, that's what we call an mRNA molecule. Why? Because it's an RNA molecule, and M stands for a message, a messenger molecule, okay? So this guy, this now what's going to happen now is this, okay? So if we go back to our next step, okay, there it is. This guy right here, essentially, if you guys can imagine it, I'll take a picture of it. All right, here we go. Let's take a picture of it. Now this guy, okay, is basically, check it out. Let's go back to our drawing here of our thing here. Okay, all right. So essentially what we've done is the following. Hold on. So essentially what we've done, guys, is by opening up this molecule, now what we've done is we've taken and we've copied this side, okay? So basically, okay, I know it might be hard to see on your screens here, but basically what we did is we used one side of the DNA molecule to help us make a copy of the other side, okay? Let me repeat that again. We used one side of the DNA molecule to make a copy of the other side. This process of copying the other side of the DNA is called transcription. So now what you guys can see, and look, and don't miss this now, don't miss this. Now this thing here, okay, what is this thing called again? Somebody tell me again. What is this thing that we made called? This thing right here in the middle? What's it called?
Chromosome.
No.
mRNA.
mRNA, very good. This molecule that we just made, the mRNA, now this guy, okay, can go out of the nucleus to the ribosome. And now what the ribosome is going to do is he's going to read it, and once he reads it, okay, he's going to be able, okay, to make the protein. That's what happened. So basically what we did right now, what we did is we did our through our lab exercise, what we did now is, okay, we have made our mRNA. So in here, this is what you guys, so right here, what you guys are looking at, okay, right here is basically the following. Check it out. Basically what you have is this diagram right here now is like we're here. So this thing went out, and now what do you think that thing on the background is? What do you think this thing in the background is over here? What do you think that is? This thing right here. This purple thing in the background. What do you think that is right there?
A nucleus.
Not a nucleus, and not a protein. What do you think it is?
A ribosome.
Exactly, it's a ribosome. Exactly, very good. So let me cancel this. So this thing in the background, right? This thing right here, this and this. In college, I always got like weirded out because like I'm like, why is he pointing at the blob? What's the blob? Like, but the blob in the background, that's the ribosome. Okay? And this thing right here that I'm circling in green, that's the mRNA. Okay? That's what you guys need to understand there. So what happened in our example so far is that, let me see here. What happened in our example so far is that our mRNA that was made, right? Oops, let's put it here. Our mRNA that was made in the nucleus now can go to the ribosome, okay? And the ribosome now is going to read and translate this mRNA molecule, okay? So let me show you what that looks like in the video, all right? So in this video, guys, I'm gonna show you those steps, okay? So now,
in human.
so here's the nucleus. In the nucleus, there's the DNA. Okay, so here's the DNA that we're talking about. Okay, DNA is super long, so that's why it looks like that. But remember, the cell only needs to copy certain portions of the DNA at a time. Okay, and so let's imagine that that piece of DNA right there is that piece of DNA that we were working with in our lab. Okay, so imagine that this piece of DNA was this piece of DNA. Okay, so let me repeat that. Okay, here we go. Let's put them together. This piece of DNA right here. Okay, screen. This piece of DNA right here is the same thing as that right there. So what did we do to this DNA again? Somebody tell me. In order to make the RNA, remember the goal is to use this. The goal is to use this DNA and get to the RNA. So what was step one that we did?
RNA polymerase.
Yes, RNA polymerase had to do, well, what do we have to do with the DNA?
Split it.
Split it, very good. So if you look at the video, right? Check it out. That's what's gonna happen right now. That piece of DNA is going to get split just like that, right? And when it gets split, see how it got split? These are the nucleotides that are being used to make the mRNA. So this thing that we're making is basically the same thing as the other side, and we're using the other one side to make the copy of the other side. So this is what I mean by transcription, copying the DNA. So if you know your vocab, then you're good, right? So now this mRNA, this thing right here, now is basically in our little activity here, right? Let me see here. So show you. So this right here, this right here that we made, right here, the thing that we made right here, right? That is that. So what needs to happen now, okay, to this thing right here? What needs to happen to this right here? To this mRNA, where is it gonna go? Where does it need to go?
To the ribosome.
Exactly, very good. So now what you're gonna see is, just like I'm showing you here, this thing is gonna leave the nucleus and go to the ribosome, all right? And so here it is, check it out. Let me speed it up a little bit. See how it's leaving? It left the nucleus and boom. See how the ribosome just got a hold of it? Does that make sense? Now what you guys are about to see now, okay, is a new process. Now the ribosome here, this guy, this big thing right here, that's the ribosome. This thing right here is the mRNA. And so now what the construction worker has to do, basically remember, think of a ribosome like a construction worker. What he has to do now is he has to build the what, Daryl?
Protein.
The protein, that's right. So this guy right here, the construction worker, is going to build the protein by using this mRNA as the instructions. This guy right here are the instructions. It's a copy of the DNA instructions, right? And so that's what's going to happen. So now take a look. See this guy right here that looks like a four-leaf clover? That guy is the tRNA, and the thing on the top is the amino acid. Because remember, proteins are made up of amino acids. So what ends up happening is the ribosome like accepts all these tRNAs, okay? And then what ends up happening is, see that thing right there that just got made? Boom.
Please.
Okay, here we go. What the heck is that? Okay, now, so this thing right here, what do you guys think that is right there? What do you guys think that is? I'll rewind it, look. Okay, check it out. Oh, we're on a little bit more. Here we go. So remember, look what happened. Boom, 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 boom. See that purple necklace thing? That's the thing that's being made. That's what the ribosome is making. He's connecting those molecules of amino acids. This thing right here is the protein, right? So now let's go back to your virtual lab and let's make a protein, all right? So let me go ahead and release it here. Let's get to that point. So everybody should be at, so now I have my mRNA. Okay, let's continue. So there's the mRNA. Now it's at the ribosome. And remember what these four-leaf clovers are. These four-leaf clovers, okay, each one of these is a tRNA. Basically, think of it like a little taxi. And what do taxis do, Aaron? What do taxis do?
They drive you places.
They drive you, or they transport you, right? That's what the T stands for, transport molecule. So these tRNAs, what they're going to do is they're going to transport amino acids. And so each one of these guys is an amino acid. Okay? And so what these tRNAs are going to do is they're going to transport the amino acids to the ribosome, and then the ribosome is going to connect the amino acids together, and that's, remember what a protein is. It's just a chain of amino acids. So now here we go. Let's go ahead and take a quick 2-minute break to kind of digest this real quick. All right. So now let's go ahead and see how this works, okay? So now, how do we read the mRNA? Who remembers? Who remembers how we read the genetic code? Who remembers? I taught this to you guys already. Daryl, how do we read it?
What do you mean how we read it?
Exactly, right? So remember, look at, basically there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 nucleotides. Think of these nucleotides like letters. Each one of them is a letter in the alphabet. So how do we read the genetic code? Do we read it 1 letter at a time, 2 letters at a time, 3 letters at a time? How do we read it?
3.
Exactly. We read it 3 letters at a time. And so that's what the ribosome is going to do. So now which of these 3-letter codes on the right matches the first 3 letters?
The second one.
The second one. So what you're going to do is I want you guys to drag the second one and see how they match. So that's basically like somebody going so basically what this tRNA did is it transported, remember what I'm talking about here, okay? This right there, that's the amino acid. That's the thing that matters. The tRNA is just the transport molecule that brings it to the amino acids. That amino acid right there, let's call it amino acid 1. That guy, he doesn't get here without this green thing. But and this code helps the ribosome know that he has the right amino acid there. So now, what's the next amino acid that I need? Which of these taxis are going to come in? Is going to come in next? Has to come in next.
The last one.
The last one, right? So when we put that one there, that matches. And look what happened to the 2 amino acids. Look. What happened to this amino acid and this amino acid? What happened? These are two amino acids. What did the ribosome do? I know you can't see it, but the ribosome actually did this. Okay, let's imagine he had hands, okay? And let's give him a face and like, boom, right? So what did he do? What did the ribosome do with these 2 amino acids?
Connect.
What did he do?
He connected them.
He connected them, exactly, right? So that's what he did, okay? Because that's his job, to connect ribosomes. Okay, this guy right here. What they do is they connect amino acids. And by connecting amino acids, right, that's how they make the protein. But there's millions of different ways to connect amino acids. How do they know how to connect them? Well, they use these instructions here that they got. So we continue with our example here. Okay, this would be the next tRNA. And look, now there's 3 amino acids connected together. Okay, now we have 3 amino acids connected together. And so these 3 connected amino acids, okay, those things, that is the protein. And so remember what I told you guys. Remember yesterday, Evelyn, what did my parents get yesterday?
They got a ride.
What did they get yesterday? What did my parents get yesterday?
They got a ride from you.
Yes, but why? Why did I give them a ride? Where did I give them a ride to?
To get a shot?
To get a shot, to get their vaccines. So basically, this is what happened, okay? And I'm gonna put this all so that it makes sense right now. So focus, look. So my parents, right? Yesterday, I took them to get their shots. All right? And so when the doctor gave them that shot, guess what was in that? Guess what was in that shot? Guess.
Proteins?
Not a protein. It was this. This thing right here. Right there. That what was in the vaccine. And so now this thing went into my mom and dad and in your parents, and it's going to go eventually into your parents as well. Now, guess what the cells of your parents, of my parents can make now?
Protein.
Exactly. They can make this now. This right here. The protein, exactly. And so now my parents in their cells, now they can make lots of these. And guess what these proteins are going to do? Okay, what do you think these proteins are going to do?
They're gonna help keep them alive.
Well, they're going to help keep my parents alive. How? What do you think they're going to do? These proteins are going to help destroy the virus. So if the virus, like let's draw a virus here. So if there's a virus that went into my parents, now my parents have the tools, okay, the ammunition, the proteins that are going to attack the virus and kill it. You see what I'm saying? That's how this works. But if it wasn't for this mRNA in the vaccine, then my parents wouldn't be able to make these proteins, and therefore that's why, okay, a lot of people are dying because a lot of people can't make these proteins. Does this make sense, yes or no? Hopefully it does. If it doesn't, don't worry, I recorded the lecture so you can watch it over again. Okay. Here. So now that the protein is made, look, there's only one code left. UAG. So now what's going to happen is there's this other little molecule that gets attached, and that tells the ribosome, hey, bro, you're done. You've been successful, and you made the protein. You're good, bro. You're done. You did your job. And now this protein goes out into the cell and does some sort of job. Different proteins do different jobs. That's why I told you, think of proteins like apps, right? And the code determines what app or what protein you're going to make and what job it's going to do for your cells. So that's what's happening inside our bodies every single day. Okay, every single day. Like, look at my hair. My hair is black, or actually very straight. But look at Annalise's hair. Annalise's hair is very curly because she is making a different type of hair protein than I am, because she has different DNA, right, that's sending different instructions. So therefore, okay, she's making different hair protein than I am, right? Okay, me and my skin is really light skin. Look at what's his name, Aaron. He's really light. We probably have a very similar protein or amount of protein that we're producing, okay, because of the DNA that we have. So ultimately, that's the trick here. Any questions, concerns? Any questions, concerns? All right. So now what I want to do is I want to make sure that you guys understand like the big picture. Okay. So now let's just recap. What is this right here? What's that?
Protein.
A protein. Good. All right. Write that down in your notes. So that's a protein. What's this right here? Right here. What's that? What's that? What was this?
A piece of DNA.
It was not. It's a piece of mRNA. Good. It's a copy of the DNA. What do we call this? What was this thing called?
mRNA.
mRNA. Very good. Okay. So here it is. All right. And remember how. And give me a thumbs up if you remember how we made it. Give me a thumbs up if you remember how we made it. Remember we had to open the DNA, copy it. Good. Okay. So now knowing these two things, okay, I'm going to teach you how to do these problems. Okay. So now, in order to make a protein, so remember our goal is to make a protein. That's the whole point of cells. The whole point of cells is to make a protein, right? So remember, okay, let me see what color is going to be cool for this. Okay, let's use yellow maybe. Okay, so imagine that this DNA was the DNA in the nucleus. So this right here, what we're going to write in this, what you guys are going to type in this column, okay, that is like us making this right there. So when you see an A, remember there's an enzyme called RNA polymerase. What's he going to put right here?
It's going to put uracil.
Uracil, very good. And then what?
Another uracil.
Good. And then?
Another one.
Good. And now it sees a T. What's it going to put here?
Adenine.
Good. Here?
Adenine.
Good. Here?
Adenine.
Good. Here?
And cytosine.
Good. Here?
Guanine.
Guanine. Here?
And uracil.
Uracil, very good.
Guanine.
Guanine.
Cytosine.
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 Guanine.
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 Cytosine.
Cytosine.
Guanine.
Guanine.
And cytosine.
And cytosine. And then?
Guanine.
Good. All right. So hopefully my C's look like G's. So now what Aaron just made, imagine Aaron right now. Whoops. Can't see that. Imagine Aaron. He was RNA polymerase. Okay. And what he did is he just made this guy right here. The mRNA. That's what he did. Okay. But obviously, how come this one doesn't look like this one? How come the one he made doesn't look like the one that from our virtual lab? How come this one, the first three are UUU, and over here it's AUG? That's what I mean. How come this one is AUG, and over here we have UUU? How come the sequence is different?
Because the first DNA strand is different.
Exactly, because we're copying different pieces of DNA. Very good. So now remember, are we done? Is that what we want to do? Is that what cells want to do? Just make mRNA and call it a day? No. What's the whole point of how do cells stay alive? By making what?
Proteins.
Protein. So what do the cells need to make a protein? They need a ribosome. Okay, so you guys are going to be the ribosome. And they need the mRNA. So we got, you guys are the ribosome. We got the mRNA, check. And they need these guys right here. What are these guys again? What is each? No, the whole thing's a protein. What is each one of these things?
Amino acid.
Very good. See that? So these guys right there, those are amino acids. Okay? And so on the test, so now what you guys are going to do is you're going to read this mRNA and become the ribosome. Right? And so now here's how we're going to do it. UUU tells you that you need, look at the screen, U. Okay? That means that the ribosome has to grab this amino acid right there called phenylalanine. So put a PHE, or if you can type it, okay, just if it fits, PHE, phenylalanine. So that's the first amino acid. So if we're making so this protein is going to have 3 amino acids. Our protein is going to have 1, 2, 3, 4, 5, 6 amino acids. Okay. So now AAU. What is AAU going to give you? What is AAU? I mean, no, actually not AAU. AAA. That's what we're looking for. So what's AAA? What amino acid are you supposed to add?
Lysine.
Okay, so A. Very good. It's lysine. So in here, in your document, type in lysine. And so what the ribosome would do is it would take these 2 amino acids and connect them together. So now CGU. What's CGU now? What amino acid is the ribosome going to connect? So this is the code CGU. It's telling them, hey, bro, you better connect. You better add CGU. Arginine. So now we added a 3rd amino acid to our protein. So now our protein kind of looks like this. It has 3 amino acids. But we have more codes that we have to read because we're building a bigger protein, okay? So GCC. Okay, that means that the ribosome has to add alanine and connect these 3 to that. The next one is GGC. Who can tell me what we're going to need? What amino acid the ribosome is going to add?
Glycine.
Glycine. So let's check it out. GGC. Very good. Glycine. There it is. Glycine. All right. And finally, the last code, GCG. What amino acid is he gonna add?
Alanine?
Okay, let's check it out. GCG. Very good. Another alanine. Very good. And so now this right here, what you guys just did is basically build the protein, except instead of having 3 little pearls, Okay, it has 1, 2, 3, 4, 5, 6 pearls. So I would have to add, okay, 3 more. And so now that's basically what the ribosome would build. It would build this protein with using these amino acids in this particular order. And this protein is going to have some sort of job out in the cell to keep the cell alive. Does that make sense? So so basically what we did here in these problems is we went through the whole process, but now I can give you problems. So if I give you the DNA and I say, okay, do transcription, then you should know what I mean by do transcription. That means copy the DNA. And if I give you mRNA and I say do translation, okay, that means, hey, bro, read the codes and tell me what amino acids are going to be in the protein. Does that make sense? Okay, so for homework, these problems right here are going to be both on the test tomorrow. So you guys are going to have to work out these problems and figure out what the answers are, so that way you can answer them correctly tomorrow during the test. And just to give you a recap, I'll do the first one for you guys, just the first portion. So remember, the transcription, you have to get your, take the DNA and make RNA out of it. So AU, CG, CC, or CG, I'm sorry. So the first amino acid would be U, G, G. So then the first amino acid would be tryptophan, right? And then you just keep doing that process, okay? So to show me that you know how to copy the DNA, right? That's step number 1. Copy the DNA into RNA. And step number 2 is, can you take this code and can you translate the code with your table? I will always give you this table over here on the right. Does that make sense? So hopefully this makes sense to you guys a little bit. A big process to digest. But like I said, you guys have access to this document. And if you guys don't have access to this document, email me and then I will give you access to it. But you guys, okay, just work through these problems and tomorrow we'll see if you really understood what we're talking about. Okay.